Good afternoon, everybody. Welcome to this webinar by Takara Bio. And the title of today's webinar is Immunogenomics to Accelerate Immunotherapy, New Solutions for T-cell and B-cell Receptor Profiling. So today we will have two speakers, myself, Mathieu Pesan. I'm NGS Product Manager at Takara Bio Europe. And I'm very honored also to have Chuang Li, R&D scientist from Takara Bio USA, who will take care of the second part of the presentation. So the webinar should last around 30-35 minutes. Whenever you have questions, you can ask them typing in the question box, and we will take some time at the end of the presentation to answer your question. So just to be all on the same page, a very brief definition of immunotherapy and the main scopes, so to say. So immunotherapy is a therapeutic strategy that is harnessing the immune systems. Of course, immune cells can be deleterious in certain diseases. They can be beneficial. So depending on the type of disease, either activation or repression of the immune cells can be sought after. So in the case of, for example, infections, bacterial or viral infections, as well as cancer, most likely immunotherapy strategies will aim at reactivating or activating the immune cell, and it's actually the opposite in some autoimmune disease, for example type 1 diabetes, where here repression might be beneficial. So there are a wide varieties of ways for immunotherapy, so a wide array of therapeutic agents. So they can be biological or chemical agents, like, for example, monoclonal antibodies. They can be antibody drug conjugates that we call ADCs. They can be cells directly, for example, T-cells. And taking cancer as a paradigm for immunotherapy research. So it is very well known that cancer has a strong immune component, and actually the tumors are very rich in immune cells. Those are mainly called tumor infiltrating lymphocytes, TILs, but there are many other type of cells, macrophages, dendritic cells, so which represent the innate part of the immune system. So as I said, tumor infiltrating lymphocytes are many T cells and B cells, which on the contrary represent the adaptive arm of the immune system. So how those immune cells are working in the tumor? So basically, the tumor microenvironment is an immunoediting milieu. Actually, tumor cells adapt to escape immune surveillance. Often there's a loss of immunogenic antigens as well, so that's a way of escaping immunosurveillance. There is also a separation of TILs homing to the tumor, and also lytic activity. And as far as B cells are concerned, there is also a suppression of antigen-specific B cell proliferation and or production of antibodies. You are all familiar with adoptive T-cell therapy. This is really linked also to CAR T therapy. So the picture depicts basically the workflow for adoptive T-cell therapy. So different approaches for T-cell therapy. One can make use of tumor infiltrating lymphocytes, so T-cell and B-cells infiltrating the tumor. But the drawback of this approach is that the culture of tumor infiltrating lymphocytes is very long to get a good expansion of these tumor infiltrating lymphocytes. So two other approaches are becoming quite popular now. They are taking advantage of PBMCs as starting material, and this is what is depicted here. Two different kind of therapies, starting with PBMCs, the so-called TCR therapy and CAR T therapy. For both of them, the first step will be first activation of the purified T cells that are drawn from the patient, so the own T cell from the patient. So activation and expansion will be needed, and for this you can take advantage of our solution, for example, the Lympho-1 medium, as well as the reagent retronectin and anti-CD3 antibody. So once the pool of activated T cells is satisfactory, of course, TCR and CAR T would imply genetic engineering. So in order to produce those modified TCRs and T cells. And for that, various tools can be used from Takara. So the classical way of doing that, as you can see here, will be to use cloning for TCR or for chimeric antigen receptor. This also implies transduction of the clone molecules by retroviral or lentiviral delivery, and here retronectin can be used as a very potent transduction enhancer. So this is the first classical way of doing that, but then of course there might be some refinements of cell engineering, and this can be done via CRISPR-Cas9 gene editing. So this encompasses targeted CAR integration, knockout of specific genes, and footprint-free engineering. And we also provide some solutions to perform that. Finally, after that, the T-cell, the modified T-cell need to be expanded again, and Lympho-1 and retronectin as well as antibodies can be used for that again. And after quality control, finally, those engineered cells can be reinfused into the patient. So we saw various approaches for immune therapy, taking cancer as a paradigm, and discussed about T cells and B cells. So it is quite obvious that those two cell types are really target of choice to design immunotherapies for various diseases. So a better understanding of T and B cell identity and function is really crucial for various diseases, so being cancer as we discussed, but other diseases, infectious diseases, autoimmune diseases. And so a better understanding of these cells will ultimately lead to the development of tailored therapy, like antibodies targeting tumor antigens, that checkpoint inhibitors, or cell therapy as discussed. And there's a growing body of evidence that, of course, T cells infiltrating the tumor are a key component of the immune system in the tumor bed, but also B cells are heavily infiltrating the tumor and might be considered as new targets. And this is nicely reviewed in that paper here. So how to take advantage of those cells, actually by knowing them better, and more precisely by focusing on their receptors. So TCR for T-cell receptor and BCR for B-cell receptor. So it's very well known that those receptors are very diverse, because they need to cope with billions of different antigens. And this is how is achieved this diversity. This is summarized in the top right side of, in the bottom right side, sorry, of that slide. So this is by the so-called VDJ recombination, where various gene segments of the TCR and BCR as well chains will get rearranged, finally transcribed and spliced to give rise to the mRNA molecule of the various TCR and BCR chains. So the collection of the various recombination derived chains are called repertoires, and this is actually what is of importance for that webinar today. So just summarizing on this slide the structure of T-cell receptors on the left and BCR receptors on the right. This is very schematic, but both receptors are heterodimer, present at the membrane of T cell and B cell respectively. And the region really of importance for us is the one highlighted in red here. This is the variable region of TCR and BCR receptors, and those are the regions responsible for recognition of the antigen that will then lead to cellular immunity for T cell, and for B cell that will lead to the production of specific antibodies by plasma cells. So how those receptors and this variability can be studied? There are many ways, actually. Historically, on the right of this slide, researchers have chosen to study TCR and BCR diversity by PCR, either starting from RNA or DNA molecules. So the drawback here is that there's the need of an a priori knowledge of those sequences in order to, of course, design primers to every single V region and to amplify this variable VDJ region. So, of course, doing so by PCR, this is susceptible to PCR bias, and it's also difficult to amplify various TCR and BCR chains in the same reaction. So that's why our preferred method, and this is what we've been developing at Takara Bio, is the strategy rather on the left, the smarter TCR and BCR profiling strategy, which is a 5' race based full-length cDNA amplification that is using template switching. We will see that in a second in the next slide, and this is followed by 2 rounds of nested PCR. So since RNA is used here as a template, we don't amplify non-expressed genes, of course, and the smart reaction combined with the 5' race will allow for sensitive detection of VDJ sequences from TCR and BCR, and will also give you the opportunity to generate full-length NGS library of these variable regions. So currently, our offering in terms of TCR and BCR repertoire profiling is the following. We have tools for human TCR profiling. So this can be done from single cells or for bulk profiling. For mouse samples, we have two kits, one for bulk profiling of TCR and one for bulk profiling of BCR. So all these kits are producing libraries with information on the complete VDJ sequence. For TCR, alpha and beta chains, subunits of TCR can be obtained, and for the mouse BCR profiling kit, IgG heavy and light chains can be obtained. All the kits are very flexible in terms of input, can start from low input amount of RNA as low as 10 nanogram up to 3 microgram, and those kits can work as well with purified cells directly as input, and in the end you will produce Illumina ready sequencing libraries of full-length VDJ. So I just told you that we chose to incorporate the SMART technology in our TCR and BCR repertoire profiling kit. This is very briefly how the SMART technology works and how it leads to full-length information of VDJ region. So starting with RNA as the template, we first perform the first-strand cDNS synthesis, where the reverse transcriptase has the ability to add non-templated nucleotides when it reaches the 5' end of the template. So this is schematized here with those X's. So you then add what we call a template switching oligo. This is the 5' oligo shown here that will hybridize to these non-templated nucleotides. And then the reverse transcriptase has this peculiarity to use the TSO as a template and add additional nucleotides which are complementary to the TSO. Doing so just by using PCR primers, we synthesize the complementary second cDNA strand and amplify the produced library. Why it does produce full-length cDNA just by definition? So this is the peculiarity of the reverse transcriptase that we are using. So this template switching mechanism is only achieved when physically the reverse transcriptase reaches the 5' terminus of the transcript. Hence the double-strand cDNA full length that you are able to produce. So as discussed already, those 4 profiling kits listed here use this technology: human single-cell TCR, bulk human TCR, bulk mouse TCR, and bulk mouse BCR profiling kits. And we just newly released this human kit for profiling BCR. This is the smarter human BCR IgG, IgM heavy and light chain profiling kit. And on top of that, we also offer you the possibility to analyze your sequencing data with the software that we released, and this is the Takara Bio Immune Profiler software. So I will now hand over to Chuang Li, R&D scientist at Takara Bio USA, and he will really give you a flavor of how works this new smarter human BCR profiling kit along with the new software. So Chuang, welcome.
Okay, thank you, Matthew. Hello, everyone. This is Chang. Welcome to the webinar. Like Matthew just mentioned, the Smarter Human BCR Profiling Kit is the newest member of our Takara's immune profiling product line. One of the greatest new features of this kit is that it contains the UMI, the unique molecular identifier. This will allow the customers to perform more accurate analysis and along with many other benefits. This kit was launched in September. At the same time, we also launched a software package called the Immune Profiler to help customers perform UMI-based data analysis. As we know, data analysis sometimes could be very tricky and difficulties, especially if you want to utilize the UMIs. So this time, we hope our new human BCR profiling kit plus the Immunoprofiler software could provide customer a complete solution for their BCR repertoire study. So in this section, I'm going to introduce you the new smarter human BCR profiling kit and the new Immunoprofiler software. So first is the workflow of our kit. So like many other Takara's NGS kit, the new human BCR profiling kit leverages on Takara's smarter technology. So Matthew just gave a very good introduction of this technology. So very briefly, basically we add in addition to the general universal adapter, we also add a UMI into the 5' of the cDNA transcripts. And in the first strain, in the first PCR, we used gene-specific primer on the one side and a universal forward primer on the other side to amplify the BCR transcripts that were interested. Then through the second run PCR, which is a semi-nefted PCR, we enriched the BCR transcripts. And because of the unique design of our two sets of gene-specific primers, we were able to capture the full length of the BCR transcripts that were interested. In current version of this BCR profiling kit, we wanted to focus on IgG and IgM, which we believe are highly expressed in the human peripheral blood circulation system and will provide more clinical relevant information. So in this kit, we provide 4 sets of primers to amplify IgG, IgM heavy chains, and the kappa and the lambda light chains. So here is the final look of our BCR libraries. We recommend customers to use 2x300 MySeq run to recover the full length of the VDJ region to get the best performance. And as you can see on the read-to side, the first 12 cycles belong to the UMI. And after that, we have a 4-nucleotide linker that we use to separate the UMI and the template switching nucleotide. So as you can see, this is really a relatively simple workflow, include only 1 RT reaction and 2 rounds of PCR with a bit cleanup in the end of the procedure. So relatively, this entire protocol can be done in 4 hours. Besides the simple workflow, the smart human BCR profiling kit also provides very consistent and reliable performance. The figure on the left side, I'm showing here, summarized a simple experiment that we tested our kit with 1 nanogram, 10 nanogram, and 100 nanogram purified human B-cell RNA, and the number of identified clonal type counts are listed on the figure. So as you can see, the increase of the clonal type count from 1 nanogram to 10 nanogram to 100 nanogram are quite consistent with the increased rate of the RNA input amount that we used in the experiment. So look at those numbers. Basically, when we use 10 times more of RNA, we roughly obtained 10 times more of clones, and it's all the way from 9 nanogram to the 100 nanogram. So this really demonstrated our kit's capability to handle samples with a wide range of complexity. As we know, humans' antibody repertoire is very complex. It changes from time to time during the immunology responses, and they're totally different from people to people. So I think that this type of capability to handle the complex samples is really what a customer needs for their immunoprofiling studies. And also, where new PCR kit showed very high reproducibilities. The plot on the right side tells us the correlation of overlapped clones between 2 technical replicates that is very high. So it basically demonstrates our kit also have a high reproducibility. So on these slides, I'm showing you is a cell line RNA spiking experiment. So what we did in this experiment is that we mixed 4 different cell line RNAs and spiked them into a 100 nanogram PBMC RNA background at different spiking levels. So the spiking levels are 1 picogram, 10 picogram, 100 picogram, and 1 nanogram. So then we made PCR profiling libraries from this all 4 spiking level samples. And because those 4 cell lines are B-cell derived carcinoma cell lines, so we know their sequences, we can actually find those clones from our sequence libraries. So the number I'm showing you in the table right here, those are the actual detected reads that belong to each cell line. So as you can see, all 4 cell lines, although they express at different levels, they can be all finded and detected at 1 picogram spiking level in the background of 100 nanogram human PBMC RNAs. So which is a very good sensitivity for our kit. So I know some of you might feel very suspicious about this 1 read, this 2 cell line can only detect at 1 read level. But really, what I'm going to tell you is that this 1 read is collapsed from UMI analysis. So basically, this 1 reads represent 12 original reads that been tagged with the same UMI. So through the UMI analysis, it's collapsed into 1, and this one is actually represent 18 original reads that been tagged by the same UMI. So really, when we use UMI-based error correction, even 1 read is very important and it counts. Another great things I want to point out here is that, as you might have already noticed, that when you look at those reads that we detect in the sequence, from top to the bottom, as you can see, the reduction rate is really matched the dilution rate of our spiking RNA amount. So to make this more clear, we converted this into a plot that I'm showing you here. So as you can see, all 4 cell lines show the very high correlation between the RNA spiking proportions and the actual detected frequency showing here. So this is just another great example to show you that our kit is consistent and reliable performance. Okay, so when thinking about the potential applications of our kit in the immunotherapy field, we expect our customers to use a lot of the PBMC RNA samples for their work, because simply the PBMC are very easy to be collected from either patients or the healthy donors. So we made a great effort to verify our kit's performance on the PBMC RNAs. So what I'm showing you here is a study that we collect 8 different single donors PBMC and purified RNAs, and we performed the BCR profiling experiment for all 4 chains, use 10 nanogram RNA of each. So as expected, we do see a wide range of in the number of clonal counts across the 8 samples, because that's what it is. That just change it people to people. But what the number is showing you is that really we can detect as low as like around 40 clones to as high as a little over 4,000 clones. That's basically 100-fold differences. And more interesting, during the process of analyze this data set, we find that with the help of UMI, you really don't need that much reads to provide all of them. We find that actually 100,000 reads per library is good enough to help you to sequence the 10 nanogram libraries. So what that means is that we recommend our customer to use MySeq, which can generate 25 million sequence reads per run. Besides the PBMC RNA, we also verified our kit with RNAs from many other sources. For example, our kit work very well with the whole blood RNA. So sometimes, the customer might don't have a large amount of blood sample to work with, so they cannot separate PBMCs from those small amount of RNAs. So that will be fine, because they can extract RNA directly from the whole blood and use our kit to perform their studies, and it will give you a good result as well. Also we we test other type of RNAs, such as the human bone marrow RNA and human spleen RNA. They both work very well. Okay, so I will also briefly talk about our immune profiler software, because sometimes we often heard from our customers that they really like to use our TCR BCR profiling kit because the chemistry is just excellent, and but they sometimes have difficulties of analyze the data. So during the process of developing this new BCR kit, we test a bunch of different software, and in the end we feel the combination of use Magic and the mixer it's a great combination that's to help us perform the UMI-based error corrections and also do the BCR profiling. During our development, we find these two software is written by two different computer languages, and it's completely compatible with our sequencing resulting files because the setup of our chemistry. So what we want to provide customer is a easy use and simple use strategy to do their analysis. So we developed this immune profiler software. Is to first it will prepare the MySeq sequencing result to make it ready for the Magic software pipeline. So then Magic will do its magic job to group the reads into different groups. They called molecular identifier groups, MIGs, based on their UMI sequences. This is the first step of error correction. Then after that, they will perform another two stages of MIG guided error correction, which will be to remove the PCR errors that's during the process, and also to collect the reads that share the same UMIs. So after that, this resulting collect FACTQ files can be used by the mixer, which is a very popular and powerful software that will perform the BCR profiling. So right now the software is launched together with our BCR kit. So customers are welcome to download them and test them. So I don't have time to do a demo here, but really the instruction is very clear, and you're really welcome to test it. So once we have the UMI into the BCR library, we really want to ask ourselves that really what are the benefits that the UMI can bring to the immune profiling. Based on the concept of UMI, the unique molecular identifier, we know that it will give you more accurate quantifications, and it will correct some sequence errors, which will give you a high confidence to call the clonal types. But after we tested and with the hands-on experience, we really believe another good or great benefit that UMI can provide to the immunoprofiling field is that it will help us understand the complexity of the samples, and therefore help us determine the best sequencing depths for the libraries. So what I want to show you here is that we have a library that have a UMI in it, but we do the data analysis based on UMI, which showed in blue here, and another is showed you that we don't use UMI, just use a regular profiling strategy. So the x-axis is the sequencing depth. As you can see, the same library, when we increase the sequencing depth, the more clone you will get. That's for sure, and that's true. But for the customers, when they handle the unknown samples, they have no idea what the complexity of their samples is. So they want to find the saturation point. Without UMI, you can see that when you increase your reads, you will get more clones, and it's never ends. So this, I feel it's probably very hard for the customer to distinguish whether those new clones that they they got by adding more sequencing reads is from a really sample is true, or just because you are accumulating a lot more of the PCR errors. But with the help of UMI, from those error correction steps, we can see that it actually reached saturation point at 100,000 reads. Of course, by the way, this is a 10 nanogram ppm cRNA input. So if we use more RNA, you need to look at our menu to find the best recommended sequencing depth. But here, what I'm trying to say is that you are wasting a lot of sequencing powers and the resources just to find a lot of PCR errors if you don't use the UMIs. And specifically, you can see that when we take the clonal count that we identified at this sequence depth and compare or overlap to the clonal count that we find here, it's basically the same. So what that means is that for a 10 nanogram ppm cRNA, we just need 100K reads to provide them. So that's made my earlier point very clear. So a single MySQL run that can really give you the chance to providing 250 libraries at the same time. So really, that's besides the other benefit. I think this is the most important benefit that UMIs bring to the immunoprofiling field. So I think that's all I want to talk today. In the end, I just would like to quickly recap what I've said about our new kit today. First, our new BCR profiling kit is a fast and simple protocol for customer to do their BCR profiling, and it can capture full VDJ region, and it can see UMIs, and will provide more accurate measurement and many other benefits that I just mentioned. Also, our new Immunoprofiler software pipeline make the UMI-based analysis much easier. So, Matthew, I think I will give this back to you. And
yes. Thank you, Chuang, for that very good and very interesting presentation, and for also showing the real added value of incorporating UMIs in this BCR profiling kit, and also for slightly touching the new software that we just released. So I will just switch to that very last slide, sorry, which is basically what we have on Takara Bio's website. So all the solutions that we discussed today for immunotherapy research. So the first one is of course the page on NGS products, the second one on immune profiling, and the final one is really an effort dedicated to put together the most relevant product we offer at Takara for integrated workflows for immunotherapy research. And with this, we will now take questions. So if you have questions, you can type them in the question box, and we will answer them. All right. So let's see. So maybe one first question. With this new smarter BCR, human BCR kit, can I use cells directly as input, or is it only compatible with purified RNA?
I can answer this question. So our kit at this stage is designed for the bulk use only. So it's recommended to only use purified RNAs at the input.
Okay, thank you. I see a second one. Which quality should be my starting material? So if I'm working with RNA, can it be degraded or not?
Ideally, we want to recommend people use high quality RNAs. Ideally, RAIN number is larger than 7 would be ideal. We haven't tested the degraded RNA as well. No, we haven't tested the degraded RNA yet with our kit, but the high quality RNA is always recommended.
Okay. So few questions regarding the new software. Are there some demo data available to test actually the software?
Yes. So basically, what we provide to customers is a relatively small set of BCR repertoire. So the total read is not that huge. So people, after they download the software, you can find the demo library in there and try it yourself. Then you will know everything, if everything is working fine.
Okay, thank you very much. And so another question related to analysis. I have my own analysis pipeline. When using your Smarter Human BCR profiling kit, do I need to use the software you just presented, or can I use my own analysis pipeline?
Yes, that's a very good question. So our design of the chemistry is a little bit unique. You know, we add the UMI through temperature switching. So if you have a bioinformatics team, it certainly can handle those UMIs, but they need to understand the final construction of our libraries. But in short, yes. You can use your own pipeline to do the UMI analysis. So, yeah.
Okay. Another question. Is the template B-cell RNA or PBMC total RNA? So I'm not sure. I understand that. Maybe we will answer that a bit later. I will switch to the next question. This is also related to bioinformatics. So thanks for the very nice presentation. So it's linked to UMI. MyJack expects single consensus per UMI. How do you handle the data if several consensus sequences are produced from a single UMI?
So could you repeat the question? Sorry.
Yeah, sorry, it was a bit long. So the question is, MyJack expects a single consensus per UMI. How do you handle data if several consensus sequences are derived from a single UMI? So basically, how MyJack how MyJack is taking care of those UMIs.
Right, that's a very good question. So basically, I think it's really related to the how many UMIs that you can have in the whole system, whether that your sample's complexity over the limit of the UMI can handle. Otherwise, you will not face the situation that one UMI connect to the several different consensus. So from or the experiment that we have done, we tested like very high complex samples, and we didn't find any thing wrong with the magic. And also magic give you a very nice automatically determine the UMI count that been needed to be repeated, showing in the sequencing runs, which we normally refer it the UMI threshold. So those things are being automatically done by the magic. So at this time point, if you feel or have doubt with the result, you can, you know, based on the magic result, you can adjust your samples complexity, either or adjust the sequence depth to get the best performance out of magic. So basically, what I'm trying to say is that it's hard for us or the software to determine at the first time if you know it's perfect. Saturated library or not, but you have the chance to make adjustment based on the first run of the first round's result.
Right. Thank you for the answer, Chong. And we have one final question. So I understand your new kit is developed for IgG and IgM. What if I'm interested in other immunoglobulin, like, for example, IgA? You also provide primers for IgA?
Very unfortunately, in current version of our BCR profiling kit, we focus on the IgG and the IgM heavy chain and kappa and the lambda light chain. So that's we don't have primers for IgA at this time point.
Okay, thanks a lot. So I don't see any other questions. So I would like to thank you all for your attendance, and I want to wish you a very good afternoon. And with that, we want to close this webinar. Thank you very much all.
Okay, thank you all.